everyone, my name is Ruiyan, and I'm a PhD student in the University of Hong Kong. And the title I want to present is the scTail process polyadenylation site detection and its alternative usage analysis from the RIS1 preserved 3' iRNA-seq data. And this is a flowchart of the This is a flowchart of the 3' iRNA-seq gene expression library conjunction from the text genomics. And as you can see, the length of the RIS1 is 115 base pairs, which is different from the normal length of the RIS1. The normal length of the RIS1 is around 26 to 28 base pairs. That is because that some lab or the companies sequence these actual cDNAs in the RIS1. We suppose that this actual cDNA in the RIS1 is a good material to detect the polyadenylation site. To prove that assumption, we compare the distance from the termination of the RIS1 and the RIS2 to the annotated polyadenylation site for a transcript, and we found that the RIS1 is closer to the truly polyadenylation site compared with the RIS2. And also here we show an example gene. You can see the first panel of the RIS1. The coverage of the RIS1 is closer to the end of the transcript. Based on this discovery, we developed scTail, which can detect the polyadenylation site by just using the actual cDNA in the RIS1. It includes two steps. The first step is calling peak. We first decompose the BAM file to the count value for each position, and then we do the clustering. After that, we do the filtering to remove some false positive clustering by setting several thresholds, such as clustering with the total number, and also the cluster length and the cluster density. Importantly, we also embedded a sequence-based classifier, which can remove the false positive clusters. And the second step is counting. We use a total raised 2. Here we define the fragment size as the destination of the RIS1 and the RIS2, and then we use the log normal distribution to fit the fragment size. After that, we can get an empirical distribution of the fragment size. So for the queries, we compare the likelihood of the queries with the likelihood threshold cited by the users, and then to decide keep or filter these queries. Finally, we can get a cell by polyadenylation set matrix. This is a detail of the sequence-based classifier embedded in our tools. It's just a convolutional neural network. You can see no matter in the human or the mouse, it can achieve a high performance. We also show the nucleotide distribution of the sequence at the polyadenylation site detected by the scTail. You can see this profile is a classic polyadenylation site profile, and it also includes two canonical motif patterns such as the AATA and also the poly-T. After proving the reliability of the scTail, we want to present the application of it by using 3 datasets. First is a human in-test dataset. Here we show example gene ZFAS1, and you can see you cannot think this gene is a cell marker of any cell types presented here. However, it includes two polyadenylation sites, proximal and distal, and you found that proximal polyadenylation site can be regarded as the cell type of the enterocyte. Correspondingly, the psi value of the ZFAS1 of the enterocyte is significantly higher than the remaining cell types. And the second data set is esophageal squamous cell carcinoma data set. It includes the immune cell and also the epithelial cell. It includes two conditions, normals and tumors. And we first use the scTail to detect the polyadenylation site. And then the breakthrough was used to detect the significant genes, which has the alternative polyadenylation site usage between the normal and tumor conditions. And with the right side, the genes located at the right side would like to prefer to use the proximal polyadenylation site. And interestingly, we found that in the most of the cell types, in the tumor condition, it always prefer to use the proximal PIs. And the third dataset is mouse furlap histogenesis dataset. It includes 6 development time points, and you can see here is example gene with the development progress and the polyadenylation site shifting happened. Correspondingly, the psi value also difference between among different development stages. However, at the expression level, it did not change. And to summary, scTail can detect the polyadenylation site from the actual cDNA in the rhizome. And also, the output of the scTail enables us to efficiently detect the alternative polyadenylation site usage between different single-cell populations by utilizing the breakthrough model seamlessly. And our manuscript have already submitted to the bioRxiv, and if you are interested, you can check the detail. Lastly, I want to acknowledge the instruction from my supervisor, Dr. Ruiyan Hou, and also the help from my lab mates. Thank you for your attention.
Thanks again. One quick question.
Hey, thank you. A very nice talk. So, you know, the 10x sometimes can also pick up basically A-rich regions that are somewhere in introns or so, but that are not actual poly-A sites. So basically reverse triangles just from these A-rich elements. So what's your criterion to get rid of these?
You mean the internal priming?
Yeah.
Yeah. Here actually we embedded a convolutional neural network. And here we first select the positive samples by use, which includes the TTF file, annotation file, and also some database, the true polyadenylation site. And then we use the start set of the transcript and also some intergenetic set as a negative samples, and use a convolutional neural network to remove the internal priming.
Thank you.
Thank you.